This video will demonstrate the steps required to design and order all necessary reagents for creating an insertion at a safe harbor site located in an intergenic region using the Invitrogen TrueDesign Genome Editor. After logging into Thermo Fisher Connect and accessing TrueDesign Genome Editor, you have several experiment options to choose from. For this workflow, we will choose long insertion, which allows for inserting anything between 31 base pairs to 10 kilobases of new DNA. Click Next. On the next screen, you will see 5 species to choose from. Select the host organism for your insertion experiment and click Next. You will then select chromosome locus as the input to identify your region of interest. You can also use the DNA sequence option. Click Next. Select the chromosome using the drop-down and input the genomic location of the start and end of your region of interest and click on Get DNA. The next screen will display a topology of your genomic region along the top of the screen with a zoomed-in sequence-level view below. Some upstream and downstream sequence is also displayed. TrueDesign Genome Editor also displays genetic variants present across populations in the gray bar above the DNA sequence. Click on the genetic variant nucleotide to view details such as its frequency and genomic position. It is important to pay attention to genetic variants as they can affect the efficiency and specificity of guide RNAs. To view variants in a specific population, click on the drop-down on the right. In the sequence editor, click on the desired insertion location as you would in a text document. A text entry box will appear prompting you to enter the sequence to be inserted. Paste your insertion sequence and click on OK. TrueDesign Genome Editor will run a check on the sequence to ensure it can be manufactured. If the sequence passes the check, a success message will be displayed and the edit list will be updated. You can undo the change by clicking the blue undo arrow in the edit list. If the sequence fails the check, a detailed message indicating how the sequence can be fixed will be displayed. Select the desired homology arm length from the options provided or enter a custom length between 35 to 1,000 base pairs. For this example, we will select custom length, enter 300, and click Next. A warning message indicating that the homology arm length has been changed from the default value will be displayed. Click OK to acknowledge. This step might take a few minutes while the software searches the region for available PAM sites, assesses the guide RNA designs for efficiency and specificity, and designs the donor DNA. When complete, you will see a table with 2 tabs, CRISPR and TALEN targets. Each row in the CRISPR table displays a CRISPR guide RNA design, its PAM site, its weighted score for efficiency and specificity, the number of predicted off-targets in the reference genome, the edit site distance from the insertion site, and the donor DNA required as a knock-in donor template for your genomic edits. If you wish to find off-targets in the variant genome selected on the previous page, click on Search in the Off-Targets in Variant Genome column. This search will take an additional 70 to 90 seconds. Green checkboxes indicate recommended guide RNAs based on their score. Visualize the guides on the sequence editor display by clicking anywhere in the row. Genetic variants are indicated below the guide RNA and PAM sequence to easily identify guides whose activity might be affected. Click on the number in the off-targets in the reference genome column to view more details such as the location, the probability of cutting at the off-target site, and annotations of whether the off-target site is in the exon or intron of a gene. Click on the number in the off-targets in the variant genome column to view more details about the off-target sites in the variant genome as compared to the reference genome. Off-target sites that have higher off-target activity in the variant genome are flagged by an orange warning symbol. To view the TALEN targets, click on the Talent tab of the table and similar information will be displayed for each talent pair. To select one or more guides or talents to add to your experiment, use the checkboxes in the table and click Next. The summary page will display all of the reagents needed for your genome editing experiment and give you the opportunity to add additional products to complete your workflow. For a long insertion experiment, the guide RNA, donor DNA, and the production vector for the donor are selected by default and cannot be unselected. Other required reagents include Cas9, transfection reagent, GCD or sequencing primers for verification of your edit, junction primers for verification of your insertion, and donor amplification primers to linearize your donor DNA from the plasmid. These are added to your experiment by default but can be unselected, or a different quantity may be selected from the drop-down lists. Lastly, you may add additional items such as positive and negative experimental controls. When you have completed your product selections, click on the Download Designs and Protocol to download your entire experimental design for future use before clicking on Add to Cart for easy one-step ordering of all selected reagents.